Welcome to the Step 1 Review Study Guide, and in this lesson, we learn about cell trafficking. The golgi apparatus distributes proteins and lipids from the endoplasmic reticulum, packages them in vesicles, and sends them to the plasma membrane. After a protein is synthesized, the golgi modifies N-oligosaccharides on asparagine residues, adds O-oligosaccharides to serine and threonine residues, and mannose-6-phosphate to proteins. A protein tagged with a mannose-6-phosphate will be sent to the lysosome. Endosomes will take up material from outside the cell or from the golgi. From here, endosomes act as sorting centers and will send them to either the lysosome, golgi, or membrane for further use. Eye cell disease, inclusion cell disease, also known as mucolipidosis type 2, is an inherited lysosomal storage disorder. It is an autosomal recessive disorder. It is caused by a defect in N-acetylglucosamine 1-phosphotransferase. This enzyme normally adds mannose-6-phosphate to asparagine residues on proteins that need to be sent to the lysosomes. However, because this process doesn't work properly, the proteins are sent to the plasma membrane and get secreted. The outcomes of this disease are coarse facial features, gingival hyperplasia, clouded corneas, restricted joint movement, claw-hand deformities, kyphoscoliosis, and high plasma levels of lysosomal enzymes. Eye disease is often fatal in childhood. An SRP is a signal recognition particle. When a protein is being translated in the cytosol, a signal recognition particle will transport the entire peptide ribosome complex to the rough endoplasmic reticulum by docking onto the SRP receptor. This allows the protein to be translated and simultaneously transported. If the SRP is defective or absent, then excess protein will accumulate in the cytosol. Vesicular trafficking proteins. There are three kinds of vesicular trafficking proteins. COPI is a codomer protein that coats the vesicle for retrograde transport. This will bring the vesicle from the cis golgi to the endoplasmic reticulum. COPI is another codomer that coats the vesicle for anterograde transport from the endoplasmic reticulum to the cis golgi. 2 steps forward, 1 step back. Clathrin is a protein that transports vesicles from the trans golgi to the lysosomes, or from the plasma membrane to the endosomes via receptor-mediated endocytosis. That's all for this lesson of Step 1 Review. We'll see you in the next video.